Hi, very good afternoon, dear friends. Today I am uploading the NEET Undergraduate 2026 Biology question paper. This was conducted during the month of May 3rd, and presently this examination has been cancelled because of the leak of question paper. So let us proceed with the analysis of this question paper. So the match the following is the first question that we have. So here in the column 1, genetically modified organism, so GMO. In the column 2, you just have to see which is that genetically modified organism. So the answer for this would be BT cotton. So A is 2. Thermostable DNA polymerase is Taq polymerase, or we also call it as Thermus aquaticus. So B is 3. TI plasmid, it's obtained from Agrobacterium tumefaciens. PBr322 is Escherichia coli. So these are the correct options. So if you look into the options A2, B3, C1 and D4. So this is the correct option for it. The next question is exploring molecular, genetic and species level diversity for products of economic importance is called as. The right answer is bioprospecting. Biofortification is addition of nutrients into already existing food. Biomagnification, it is accumulation of non-biodegradable substances in the higher trophic levels. Bioremediation is a remedy for pollution. So the right answer for this is bioprospecting. So bioprospecting is the right answer. So now this is an interesting question that we have. Which of the following statements are true with the reference to the sex determination in honeybees? Choose the correct answer from the options given below. An offspring formed from the union of a sperm and an egg develops as a female. So which can be queen or sterile female worker. Yes, that is the correct option. So an unfertilized egg develops as a male by parthenogenesis. Parthenogenesis is formation of organism without fertilization. B is also correct. C, a male has half the number of chromosomes than that of a female because they are haploid. C is also correct. ABC is correct. D, males produce sperms by meiosis. This is wrong because already males are haploid. They cannot undergo any meiosis. So the males produce sperms by mitosis, not meiosis. So D is the wrong option here. E, honeybees have a haplodiploid sex determination system. So A, B, C and E are correct options. So A, B, C and E are correct option. Next one is 2,4-D, match the following. Column 1, they have given 2,4-dichlorophenoxyacetic acid. So it is used as a dicot herbicide. So A3. So there is only one option of A3, that is 4th. Anyway, GA3, gibberellic acid 3 is used in brewing industry or beverage industry. Kinetin, it is utilized for nutrient mobilization. Abscisic acid, it is going to initiate the closure of stomata. Cytokinin is going to facilitate the opening of stomata. Abscisic acid, it is going to stimulate the closure of stomata. So the option A3, B1, C4, D2, fourth option is correct answer. So A3, B1, C4, D2. In Racemose inflorescence, the growth is limited. Flowers are solitary. Flowers are born in an acropetal succession. That is, older flowers are at the base and younger flowers are at the apex. We call it as acropetal succession. Main axis terminates in a flower. So here the right answer is flowers are born in an acropetal succession. Remaining sentence are all wrong. Since the origin and diversification of life on Earth, there have been 5 episodes of mass extinction of species. How is the 6th, which is in progress, different from the previous episodes? The current species extinction rates are far lower than those in previous episodes. The present species extinction rates are 100 to 1000 times faster than in the pre-human times. The present net species extinction rate is 0. The current extinction species rates are nearly 10 times faster than that in previous episodes. The correct answer for this is second one. The present species extinction rates are 100 to 1000 times faster than in the pre-human times. Alpha helix is found in which level of protein structure? It is not seen in primary structure for sure. So it is found in secondary structure. Alpha helix is found in secondary structure of protein. The enzyme required for carboxylation in the Kelvin cycle, that is fixation of carbon dioxide, is RUBP carboxylase, oxygenase, or Rubisco enzyme, we can also call it as. So the answer is RUBP carboxylase oxygenase. Arrange the following in the correct developmental sequence related to microsporogenesis. So the microsporogenesis, the correct sequence would be, it begins from D, pollen mother cells, and pollen mother cells are going to, rather than D, it can begin from B, sporogenous tissue, BD, C is BDAC. So BDAC, fourth option is correct. That is, sporogenous tissue is the first stage, pollen mother cell is the second stage, microspore tetrad is the third stage. From tetrads, they are going to form the pollen grain. That is the fourth stage. So BDAC, fourth option is correct. Which of the following statements are not true regarding restriction endonucleases? So they are called molecular scissors. These are the enzymes. So restriction endonucleases, molecular scissors are correct. These are the enzymes responsible for restricting the growth of bacteriophages in E. coli. They cut the DNA only at the center of the palindromic site. They remove nucleotides. They remove nucleotides only from the end of DNA fragments. Here we have to find out the incorrect statement. They recognize specific palindromic base pair sequences. So here the incorrect ones are, so the incorrect statements are, these are the enzymes responsible for restricting the growth. So B and C seem to be the incorrect statement. C and D, sorry. So C, B is also correct. C, they cut the DNA only at the center of the palindromic sites. It can also cut at any ends, like echo R1. They cut in between A and G, and they are not the center of the palindromic site. They result in the formation of asymmetrical ends. Then D, they remove nucleotides only from the ends of the DNA fragments. That is endonucleases. That enzyme is restriction endonuclease. Exonucleases, they do the cutting of nucleotides only at the ends. So D statement is wrong. C and D. In the lac operon, the Z gene codes for beta galactosidase. You know, in the lac operon, the structural genes ZYA. Z codes for beta galactosidase, Y for beta galactosidase permease, and beta galactosidase transacetylase. These are the three enzymes coded by the ZYA. Z codes for beta galactosidase. Fourth option is correct. Match the following. This is from the cell cycle. So G1 phase, what exactly happens here is it actual cell division occurs? Cell is metabolically active and continues. Synthesis of DNA occurs. In S phase, synthesis of DNA occurs and the amount remains the same. Proteins are synthesized while cell growth takes place. So here, actual cell division occurs in M phase, D1. So then G1 phase, So cell is metabolically active, and so the answer, it would be like A to fourth option. G1 phase cell is metabolically active and continues to undergo uh, growth. Then S phase synthesis of DNA occurs, and the amount of DNA remain and the amount of DNA doubles up. G2 phase, what you can notice is uh, the proteins are synthesized while cell growth continues. Actual cell division is M phase. So fourth option is correct. The respiratory quotient of a biomolecule used for respiration as per the above equation would be C57H11 110 O6. So there is 145 oxygen, so that is consumed, and 102 carbon dioxide. So it would be lesser than 1. So it could be somewhere between 0.5 and 0.95 approximately. That is the answer. Which one of the following is not a characteristic of plant cells in the phase of elongation? Elongation phase, like new cell wall deposition, so cell enlargement, large conspicuous nuclei, increased vacuolation. In cell elongation, uh, the third option seems to be the wrong one, because this is seen before cell division, that is not during cell elongation. Large conspicuous nuclei is the wrong option. Arrange the following steps of somatic hybridization in a correct sequence. Choose the correct answer from the options given below. Digestion of cell walls, somatic hybridization, correct option, correct answer from the options given below. So which are the correct option? We have to check it, and we have to arrange them in the proper sequence. So digestion of cell walls, isolation of naked protoplast, fusion of protoplast to get hybrid protoplast, isolation of single cells from two different varieties of plants, growing of hybrid protoplast to form a new plant. So D, isolation of single cell from two different varieties of plant, then isolation of digestion of cell walls. DA, fourth option is correct. So third option would be, the third sequence would be isolation of naked protoplast, then fusion of protoplast to get hybrid protoplast, then E is growing of hybrid protoplast to form a new plant. DABCE is the correct option of arrangement. Conjunctive tissue. So it is usually found in between the xylem and phloem of root. So that third option, A3, is correct. B, Casparian strips. It is seen in endodermal cells rich in starch. Subsidiary cells, specialized cells. So A3, B, B2, A3, Casparian strips. B4, endodermal cells with suberin deposition. Subsidiary cells, C is 1. So second option, A3, B4, C1, start sheet is nothing but endodermal cells with suberin deposition. So second one, A3, B4, C1, D2. In angiosperm, root hairs arise from which one of the following regions of the root? Region of maturation. So third one, root hair arises from region of maturation. Third option is correct. Which of the following floral formula is the correct floral formula of Solanaceae family? It will be having K5, C5, then bicarpellary. Only thing is, so K5, C5, A5, G2. The right option here is The third one, K5, because all the petals are fused, corolla is fused. Andrecium 5, they should have shown that epipetalous condition. Then gynoecium 2. So all these are correct options. A to C, that epipetalous condition would have been shown in the third one. That is the reason it is correct. Which one of the following is a triploid cell? Synergids are haploid. Primary endospermal cell, primary endosperm, PEC, it is triploid. Central cell is diploid. Zygote is diploid. So second option is triploid. Primary endosperm cell is correct one. So match the following: decomposition, detritus, mineralization, humification. Decomposition is breakdown of complex A3. Detritus, dead remnants of plants and animals. So A3, B4. So fourth option, and C2, mineralization, release of inorganic nutrients by the components. Then humification. So humification is accumulation of dark colored amorphous. So A3, B2, A3, B2, C, A3, B4, C2, and D1. So fourth option is correct. The main criteria used for 5-kingdom classification proposed by R.H. Whitaker included. So which are the correct options? So main criteria for 5-kingdom: cell structure, body organization, presence of flagellum, reproduction, phylogenetic relationships. So cell structure, body organization, A, B, D, E. Fourth option is correct. A, B, D, and E only. Presence of flagellum is not an attribute. Evil quartet of biodiversity loss includes which of the following? So there is which of the following: habitat overexploitation, alien species invasion, air pollution, co-extinction. Habitat loss and fragmentation, habitat loss and fragmentation, overexploitation, alien species invasion, co-extinction. So probably the most probable answer would be the third one. Let us see. Yes, habitat loss and fragmentation, overexploitation, alien species invasion. So they are the and co-extinction. They are the four evil quartet of biodiversity. Arrange the following steps of DNA fingerprinting in a correct sequence. So correct sequence, we have to arrange it here. So isolation of DNA and its digestion by restriction endonucleases. So for DNA fingerprinting, hybridization using a labeled VNTR probe, transferring of separated DNA fragments to synthetic membranes, detection of hybridized DNA fragments by autoradiography, separation of DNA fragments by electrophoresis. See, first one is isolation of DNA and its digestion by restriction endonucleases. E, separation of DNA fragments by electrophoresis. Then B, hybridization using a labeled VNTR probe. Then you have C, transferring of separated DNA fragments to synthetic membranes. So AE, CBD, fourth option. Isolation of DNA, E is separation, C is transferring of separated, B is hybridization, D is detection of hybridized DNA fragments by autoradiography. So fourth option is the correct one, AECBD. So which of the following statements are correct with reference to a transcription unit? So which of them are correct? Transcription unit, they have a terminator, process of transcription. So transcription is synthesis of mRNA, terminator, process of transcription. Choose the correct answer from the option given below. A transcription unit in DNA is defined primarily by three regions: promoter, structural gene, and the promoter is said to be located towards the 5' end of the structural gene. The promoter is a DNA sequence that provides binding site for RNA polymerase. The promoter defines the template and coding strands. The terminator is located towards the 3' end of the coding strand, and it defines the end of this. So correct options, which are the correct options here? The correct options are A, B, C, D, E. All of them are correct options. Which one of the following types of pollination brings genetically different types of pollen grains to the stigma? So it is going to be xenogamy. Autogamy is not going to bring about it. So cross-pollination, the answer is xenogamy. Geitonogamy is functionally cross-pollination, but genetically it is autogamy. So xenogamy is functionally and genetically both of them are cross-pollination. Which of the following is an in situ conservation method? Seed banks, ex situ is outside of it. So in situ conservation, seed banks, sacred groves, botanical gardens, wildlife safari parks. So the answer for this would be the sacred groves. Seed banks are artificial, sacred groves are dedicated forest areas. Botanical garden, wildlife safari parks, they are all ex situ method. Sacred groves are in situ, in natural methods they are. Heterophilous development in response to environment is an example of which of the following phenomena? Plasticity. Fourth one is plasticity. Productivity, net primary productivity, gross primary productivity, secondary productivity. Productivity is rate of biomass production. Net primary productivity, it is GPP minus R, net primary productivity. Rate of formation of, so net primary productivity will be 1, productivity 4, A3B1, that is the correct option. So C, gross primary productivity is rate of production of formation of so that answer would be A3B1C4. Gross primary productivity is rate of formation of new organic, rate of production of organic matter, and secondary productivity new organic matter by consumers. So A3B1C4D2. Which of the following statements are correct regarding amino acids? Choose the correct answer from the options given below. They are substituted methanes. Amino acids, yes, that is correct. Serine is an aromatic amino acid, wrong. Valine is a neutral amino acid, correct. Lysine is an acidic amino acid. So choose the correct answer from the option given below. So A, C, A and C only is the correct one. Lysine is a basic amino acid, not acidic. A and C only. Fourth option is correct. In which of the following the ovules are not enclosed by an ovary wall and remain exposed? In which of the following the ovules are not enclosed by an ovary wall and remain exposed? It is in Pinus. Pinus is Gymnosperm. Wolffia is Angiosperm. Funaria is bryophyte. Selaginella is pteridophyte. So Pinus is naked seeded plant. So Pinus is the correct answer. Which of the following statements are correct with reference to packaging of DNA helix, chromosomal proteins? Packaging of chromosomal proteins in DNA helix. Choose the correct answer from the option given below. Histones are organized to form a unit of 8 molecules called histone octamer. Histones are negatively charged basic proteins. Histones are rich in the basic amino acid residues lysine and arginine. The positively charged DNA is wrapped around the histone octamer to form nucleosome. The packaging of chromatin at higher level requires an additional set of proteins called non-histone. So choose the correct answer. So histones are organized to form a unit of 8 molecules called histone octamer. That seems to be correct. Histones are rich, A, C, E. So let us see. The correct option is A, C and E only. So marginal placentation. Marginal margins it is. So you can see that in P, A2. Axial placentation, you can see it in lemon, A2, B4. C, parietal placentation, you can see it in mustard. Basal placentation in marigold. So C2, A2, the answer should be A2, B is B4, C1, D3. First option is correct. Which one of the following is the site for active ribosomal RNA synthesis? So nucleolus is the site for active ribosomal RNA synthesis. The main function of Bulliform cells in grasses is to perform photosynthesis, minimize water loss during water stress. So the second option, they help in rolling up of leaf lamina during scarcity of water. That is, second option is the correct one, to minimize water loss during water stress. Which of the following statements are correct? In the lake, loss in the lake. Choose the correct answer from the options given below. The Amazon rainforest being cut and cleared for cultivation of soybean is an example of habitat loss. Yes, that is correct. A is correct. Stella's sea cow and passenger pigeon became extinct due to overexploitation by humans. The Nile perch introduced into Lake Victoria helped in population growth of cichlid fish in the lake. So water hyacinth is an invasive species, correct. When a species becomes extinct, the plant and animals species associated with it are not affected. So E is incorrect statement. Water hyacinth, D is correct statement. So A, example of habitat loss, A is correct. A, B and D. Let me see if the first option, yes. Stella's sea cow, A, B and D only. Nail perch introduced in Lake Victoria in East Africa helped in the population growth of cichlid fish. Helped in population growth. That is wrong. It is reduction in cichlid fish, not population growth. That is the reason A, B and D only is the correct option. Which one of the following statement is not true about the universal rule of binomial nomenclature? The first word in biological name represents the specific epithet. It is wrong. The specific epithet in the biological name starts with a small letter. So both the words when handwritten are separately underlined and printed in italics. Biological names are generally in Latin. The first option is incorrect statement. The first word in the biological name represents the specific epithet, while the second component denotes the genus is an incorrect statement. Which one of the following disorder is caused by substitution of glutamic acid by valine? So it is sickle cell anemia. At the sixth position of the beta globin chain of the hemoglobin molecule, third option, sickle cell anemia. Find the incorrect statement about photosynthesis from the following. Choose the answer from the options given below. So incorrect statements we have to find out. Water splitting complex is associated with PS1. It is incorrect. C4 plant use the C3 pathway of carbon dioxide fixation as the main biosynthetic pathway. B is also wrong. In C4 plants, photorespiration does not occur. C3 plants exhibit Kranz anatomy. That is wrong. A, B, D. ATP synthesis in chloroplasts occurs through chemiosmosis. That is correct. A, B, A and D only. The water splitting complex is associated with PS1. A and D only. Fourth option. B is correct because C4 plants uses the C3 pathway of carbon dioxide fixation for synthesis of food, whereas it is just escorting the carbon dioxide up to the from mesophyll cell to bundle sheath cells. So that is the correct statement. So A and D are incorrect statements. So here the trypsin they have given it. So it is an enzyme. Trypsin is an enzyme. Concanvaline A is a lectin. So A3, morphine. Morphine is alkaloid. A3, B4, C2, collagen, D1. So first option is correct. Identify the correct statement about biomolecules. Choose the correct answer from the options given below. Lipids are generally water soluble. It is wrong. Proteins are polypeptides. Correct. Polysaccharides are long chains of sugar. Correct. Adenine and guanine are substituted pyrimidines. Almost all enzymes are proteins. So A, B, lipids. So B, C and E are correct. Third option. Incomplete dominance. Example for this is inheritance of flower color in antirrhinum. Codominance is ABO blood group. Pleiotropy. So polygenic inheritance is human skin color. So A2, B4, A2, B4, C3, D1. Third option is correct. A3, B4, C3, D1 is the correct option. Identify the correct sequence of steps in each cycle of polymerase chain reaction. You can always remember it as DAY. Denaturation, annealing, extinction. Second one is the correct one. How many ATP and NADPH molecules are required to make one molecule of glucose through the Kelvin pathway? So one molecule of glucose, so it requires 18 ATP and 12 NADPH2. Second option is correct. Match list with one with list 2. Glycolysis, where does it occur? In the cytoplasm of the cell. ETS, electron transport system, it occurs in inner mitochondrial membrane. So A3, B1. So last option is the correct one. Accumulation of protons, it is going to take place in intermembrane space. Krebs cycle takes place in C4. A3, B1, C4, D2. Fourth option is correct. Which of the following statements are correct with respect to DNA separation, isolation and visualization? Choose the correct answer from the options given below. The cutting of DNA is done by molecular scissors. Isolation and visualization, DNA separation. The DNA fragments separate according to their size in an agarose shell upon electrophoresis. The separated DNA fragments can be seen without staining. It has to be seen with staining when exposed to UV light. It has to undergo staining. Okay. The separated DNA fragments, when stained with ethidium bromide, can be seen in visible light. That is wrong. So A and B are the correct options. First option is correct. Remaining are wrong. What is the probability of having children with O blood group where both mother and father are heterozygous for A and B blood group? So probability would be 50%, where both mother and father are heterozygous children with O blood group, so the probability is 50% would be O blood group. 25% would be O blood group. Third one, 25%. Reason for this is there is a possibility of A blood group, B blood group, AB blood group and O blood group. So all of them are in 25% possibility it is. So that is the reason it is 25%. Mollusks, what do they exhibit? Mollusks. So reptiles, they have pulmonary respiration. Adult amphibians, they can show pulmonary and cutaneous respiration. Amoeba is cellular respiration. So the reptiles, mollusks is branchial or gills respiration. A2, B1, C, A2, B1, C4, D3. Fourth option is correct one. A2, mollusks, branchial respiration, reptiles, pulmonary respiration only, adult amphibians, pulmonary and cutaneous, amoeba, cellular respiration. So fourth option is correct. Insertion of a foreign DNA at BAM-H1 site in an E. coli cloning vector PBR322 results in the loss of antibiotic resistance towards ampicillin and tetracycline, ampicillin, tetracycline. So tetracycline is the answer because BAM-H1 comes there. So tetracycline. So insertional inactivation you should know. What is the reason behind production of large holes in Swiss cheese? The production of large amount of carbon dioxide and H2 by Trichoderma polysporum, the production of large amount of carbon dioxide and hydrogen by lactic acid bacteria called lactobacillus, production of large amount of carbon dioxide. So third one is the correct action by Propionobacteria shermani. Yes, that is the